everyone can hear me?
Can you wait a bit? Let me just, we are about
okay.
we're about to go live in YouTube. It's just taking a bit.
Okay.
Eugenio, announcements for next week?
Yes. Yeah, so we will have this, so next Saturday, we're gonna launch the Public Journal Club Developmental Biology. So we also want to invite you to join to the Saturday session. We will have a great paper from Camargo Lab at Harvard University. So it's gonna, this paper which uses really single cell lineage tracing for the discovery for hematopoiesis function. So it's really a good paper. We have previously session, we have looked for some of the Camargo labs. We reviewed the Carlin mice, and today, and next Saturday, we're gonna review Alejo Rodriguez paper. So it's gonna be a great opportunity, guys. If you could join us, it will be great. And yes, keep in touch in our social networks, and please share with your colleagues, friends, and universities. We want more and more people to join this initiative.
Okay, so we are live now.
Okay, perfect.
Okay.
Thank you.
So here we go. So everyone can hear me? Okay. So the paper we are going to review today is entitled "A Novel System to Map Proteins Interaction Reveals Evolutionary Conserved Immune Evasion Pathways on Transmissible Cancers." So just to remind all of our members that we have a robust way to try to know what do you want to see on the Public Journal Club, and in order to see what is what you expect or what do you are interested to see in the Public Journal Club, we take advantage of this robust system of know if are you interested in explore other branch of immunology, and I ask you all through an Instagram story if you are interested, or when, well, first, if have you ever heard something about wide immunology or ecoimmunology? And it's 50-50% that you have actually heard something about wide immunology or ecoimmunology. And after that, we ask you if you are interested to see something about wild immunology on the Public Journal Club, and 80% say yes, we want to see something about wild immunology, and the other 20% say no, we don't want to see nothing about wild immunology. And to this day, I don't know who are these two persons, or that really don't are interested in wild immunology. But after this session, maybe you can see wild immunology or ecoimmunology in a different way. So, okay, this work is was done on the very recent funded laboratory of Andrew Flies, where, and this paper is was published on Science magazine, and it's entitled "A Novel System to Map Proteins," blah 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 blah. And also Andrew Flies have a comment article where they is reviewing some interesting things that allows to understand better what happened in the immunology, what is shaping immunology in wildlife, and actually how this could improve the research on clinical research or translational immunology research. So unless today we are going to review this paper, I would like also to encourage you to just wait a minute. I will also like to encourage you to review this this piece because it's pretty interesting. And one quote that this paper had is that no modern organism can replicate all aspects of health and disease in another species. So I think this is a pretty important thing, because we are we usually work with different animal models, and we usually try to say no, this organism is better to understand this process in human, or this model is better to understand how is actually work this process in different diseases. So this quote maybe could be hard to swallow to all of us, but after this session, I hope you can see the wide immunology and what is actually the ecology part in immunology that is too important to understand all the processes in immunology. So okay, and once again to go back to the answer we made you on Instagram, and so we say that you never have been the 50%. You say that you never heard something about wide immunology or immunology. So the other 50%, I hope, is here today. So maybe they can take talk us more something about wide immunology. So as mentioned before, Eugenio, I was studying different processes of wide immunology during my master degree, and today I would like to start with a pretty fast introduction of what is actually wide immunology. So all organisms in animal kingdom have another sense that actually led them to see or explore what is different of itself, or what is actually or what is actually different on the environment. So these processes are pretty important, and now more than ever, because we have a lot of emerging infection diseases, and these diseases have a common target molecules that can be shared among taxa, and also this can migrate to one host to other and become more dangerous to humans. And this is basically because humans, we are pretty weird and will enjoy to do something weird things like kissing wild animals. And in this way, and also because there is a rise and born of how of a kind of uh, ecotourism, and this could lead to make more make more fast the transmission of certain emerging infectious diseases. And so also, while immunology, what is looking for is how these processes are conserved among taxa, and also how this could be shaping the health of the world. So this is understand that, like, if something in the one ecosystem is good, so this could bring some benefits to the human. So, and this conception of what is better for one species is also good for another species is called One Health. So white immunology also have a different branches, but one of it is that they in white immunology is looking for One Health. What is the health for every organisms around the world? So on the paper, Andrew Flies and colleges start to telling us that there are certain cancer processes that are actually not attended by the different immunology approaches, and given that there are a lack of molecular that can target these cancer processes, and actually tell us that the human is not the only species in the animal kingdom that is actually got cancer, and also there are different checkpoints that is like certain molecules that are shaping the progress of cancer, and also they talk us a little bit about MHC. So MHC class 1 and class 2 are pretty important to white immunology because let us to understand how these processes well to monitor different processes in wildlife. Why? Because MHC, as I hope most of you know, let us to distinguish between what is your and what is not your tissues or your cells, etc. So the model that they study is devil Tasmania devil, and this Tasmania devil has a pretty interesting phenotype that they have got cancer, but they got cancer in a different way that we got cancer, humans and a lot of other species, because they get cancer by a different way, because the cancer acts uh, like a pathogen, so they can get cancer, and after that they develop different cancers, and this is basically because they can distinguish between what is their own MHCs and what is not their MHCs. So also there is this process has the main cause of the decline of population of Tasmania devil, and there are two different cancers actually known in Tasmania devil, and the first one is the devil facial tumor 1 and devil facial tumor 2. And the devil facial tumor 1 is characterized by the decrease of MHC, as I told you before, because they are not able to recognize between and distinguish between what is their own tissues or what is not their own tissues, and this led um, um, to spread this cancer as it it could be a pathogen. So in the second type of cancer of tumor of Tasmania devil, there is a certain studies that show that MHC class 1 actually is also upregulated in these processes, but also there are too many evidence that show this is also an important cause to in the Tasmania devil decline population, but also there are no a well characterized hallmark as MHC decrease in the class 1, because in the class 2 also there are expression of MHC class 1. So these two are understood like two different processes, but also they cause a decline in the devil in Tasmania devil population. So okay,
Aranzazu?
okay.
Just before you continue, I just want to tell the audience that and welcome Patrick. So Patrick Leonard is now in the session. So Patrick is also one of the authors of the paper. So Patrick, if you could, if we invite you to comment any idea regarding the paper during the discussion. Okay.
Yeah, and guys, also if you want to add something or if you have any doubt, I would like to encourage you to ask through chat, or also you can just tag up. So okay, may I continue, Eugenio?
Yeah, sure. Go on.
Okay, so okay, and well, once again, as told you before, these processes are characterized well characterized. Okay, Eugenio, I guess we have questions on the chat. Ah, okay. Welcome, Patrick.
If
Okay,
you have any
so
comments during the presentation, please let us know to have really cool paper discussion.
You can write in the chat.
yeah. So once again, this is a these cancer processes are well characterized that there are too many checkpoints characterized in human processes, but there are a lack of markers of cancer and tumor processes in wild animals. And also, this is important because wild animals could let us to know what is happening in the whole system that is shaping also by the environment and is also shaped by the different pathogens, because the the most of the Harvard laboratory animals lack of different these signals when you are testing different cancer processes, and you can really you you really don't know if this process could be uh, could be driving what is driving these processes could be also in the wildlife or well, in human common life, and how this could be interact. So okay, next. Okay, so Andrew Flies and colleges develop um, a pretty interesting and also creative way to target different molecules that are involved in this cancer and tumor processes. So in order to summarize these processes, I will explain you how this works. And basically, you have a gene of interest target, and you can take these sequences, and after that you can uh, design a expression vector and put these sequences in different in these expression vectors, and after that you can in a way that copy paste this to express a protein that also is after that transferred in a different line cells, and then you can purify it with a column, and after that you can report that this protein is actually works because they start fluoresce. So okay, well, something that I would like to point of this method that this could be works not only uh, for Transmaria debut as um, unless this all this data that I will to presenting you is also developed performed in Tasmania debut. This could be also using for different animals or different purpose as diagnostic or also therapeutic therapeutic processes. So basically, you know this works in different animals because it's limited by the characterized to the your gene of interest, and this gene of interest have to be widely conserved among taxa, and in that way you can generate a target that could work for more than one species. So one limitation of this process maybe, and to my and is my guess, is maybe that it's limited by the only genome available organisms. So maybe this could be, well, this could be no limitation, but um, this could be maybe more difficult to those animals that lack of genome annotation. So okay, and after this after this um, quality control of the system, they see that no matter what is the fluorescent protein reported, this system works, and only works when this is activated, or this is linked with the target. So they prove this because first of they link a wide spectrum of well-known target proteins that are involved in cancer processes, especially in Tasmania devil phase tumor, and this is this work well when only when the system is activated, and also when this is and also when this is the ligand is present in the system. And also they try to co-stimulating the in different in an ex vivo in an ex vivo experiment. They test if in different culture system that this also happen, and this could be led by these molecules only when the ligand is present. This led us to know that this is this system is specific, and this only when the ligand is present in the system. And
Yes, but I mean, I just want to, sorry, Aaron, I just want to comment that I really found it interesting that somehow I really I hope Patrick could help me with this, or anyone in the audience. So it seems that only they see signals if they actually put chloroquine, right? So they're actually blocking lysosomal. So they put
yeah.
yes. So this is really interesting because I don't know which I might be some confusing, but there are some experiments where they show that adding these molecules in the supernatant and without chloroquine, even somehow they bind the receptors, they don't have any signal. So that's why they put chloroquine. So we have, yes.
Yeah.
Yeah, I'm happy to chime in if you guys like. Yeah, so effectively we found that there wasn't really any signal when there really should have been, and a couple of the other members of our lab were able to dig around in the literature and found that some of these receptors, particularly when you look into CTLA4 and such, they actually end up being taken from one cell to the other and uptaken and transendocytosed. So we investigated that a little bit and found that we could block that using that drug. And when we did, we actually managed to restore some of the binding. So it was that was quite the breakthrough for the paper. If we're joined by Peter or Andy, they'll probably be able to brief us a little bit further on that. It wasn't quite my department, but I do remember when we finally cracked that.
Thank you, Patrick.
Thank you. So, okay, if
Excuse me,
okay,
may I ask something?
yeah, for sure.
Well, what I understand of this system is that the idea is to fuse the ligand of the protein of interest with a reporter, a fluorescent protein, and so we can detect, for example, CD86 by joining it to CTLA4. But what I don't understand is what is it necessary with CTLA4 and CD80 to add this FC tag? I don't understand why is it necessary.
I can take that one again if you like.
Okay.
Yeah.
Yeah.
Yeah. Essentially, it's essentially a screening tool. So for a lot of these receptors, they also have multiple binding partners. They don't just bind to their exact opposite. So you do need to screen a bunch of these different receptors to be able to make sure which interactions that we know of in humans or mice are being replicated in the devil. Yeah, that's essentially why we screened all of these different ones, in short.
Okay, thank you.
Okay, so if there are no more questions about this slide, so I will.
Yeah, I mean, I'm sorry. It's
Okay.
really a naive question, so it maybe have no fundament, but I don't know if these molecules, when binding to the receptor, could they like activate the receptor or give any signal at all? This is like a really naive question.
No, not at all. So in the native cells, they are very likely to transduce a signal. But in our model system, in essentially hamster cells, they're a lot less likely to conduce the same signal, or it might be a different signaling pathway that they work through. So you're unlikely to get the exact replication. This is more to see just the receptors and the ligands at the cell surface and be able to detect them like rapidly. But in terms of sort of investigating the downstream functions, it's a little bit more difficult to tell, particularly when you're just working in the hamster cells, not really devil immune cells or cancer cells specifically. Does that make sense?
Yes.
Cool.
Okay. So, okay. In this part, they found that actually CD200 is upregulated in different devil phase tumor, and also they found that in these tumors, and the protein level is this could be maybe not replicable because in the tumor, in the white tumors, they can see a signal of CD200R1 and CD200 lion. So under the reported of orange, but after that, when they try with a different driver or promoter, they can see this this signal of the CD200 proteins and CD200R1 and CD200 protein is also reported under this different driver. So, and in this part also they have one pretty important insight that when the in a system that where is overexpressed CD200R1, the CD200 surface expression is black. So and some in and somehow this this CD200R1 is blocking the signal of CD200 protein. That is pretty interesting because after that they discuss they say some pretty interesting things about this, how this pathway could be regulating actually different cancer processes among tags, and how this could be also well conserved also in the among animal kingdom, and different ways that they could be important to the treatment of different cancer process. Well, I don't know if okay, well, okay. So also they see that these proteins could be also upregulated in different tissues, but the main tissues that are upregulating this pathway are the facial tumor 1 and tumor 2. So if anyone want to add something or add something about this slide.
Yes. I'm curious about the so it's really interesting that even though they found these molecules that are overexpressed, and when they did in this cell line, they didn't see but you never see a signal until you overexpress the protein. So I don't know if you try to do this same experiment, maybe thinking in another type of protein that is really highly expressed in the cell, that could actually see a signal without overexpressing the protein.
Yeah, so for a lot of the different molecules, we did initially screen them against, say, DFT cells, generally working in the first devil line. So yeah, unfortunately, a lot of that is just sort of preliminary, so it hasn't been included here. But yeah, so we do some, but then we also had some problems in terms of the signals that we were getting. So depending on what tag you have as part of your construct, that might confuse the signal, depending upon all the other things that the devil cells are expressing. So yeah, so this is essentially just the model system to sort of delineate those receptor and ligand interactions, to sort of take away that background, if you will.
Yeah, thank you.
Okay. And well, in order to understand how this could be a hallmark of the progress of tumor and cancer, they want to, well, they thought how the blood cells could be also inside early inside of the early stages of cancer development and also tumors, and for this purpose they try to see the expression of if circulating tumor cells and and the tumors actually in the tumor cells actually in the tumor, how are shaping this the develop of these different tumor development. And they found something that is really interesting, that as I mentioned you in the start of the meeting, I tell you that the devil facial tumor 1 is well known to upregulate the MHC class 1, and the devil facial tumor 2 is, there are too many studies and too many insights that could be a hallmark of this tumor process. So what they found first is that the devil facial tumor 1 is actually express the same levels of CD 200 that that is indistinguished between the blood cells, and is also have the same level of expression. But despite that, the devil facial tumor 2 is up is up is less less is less, has different expression levels than are less than DFT1 and blood cells. This is this signal could be distinguished between the blood cells and the tumor cells in the tumor. So this could be an early hallmark of the development of cancer processes, and maybe could let us also know how this different progression of malignity of also processes of tumor development could be mm, could be diagnosticated in early stages in other species. And in order to try to prove this insight, they also prove that this is also is this system is also adaptable to different organisms. In this case, they use human to see that this system works only when the PDL1 is actually in the system, but no, and this is specific because this is not linked with in a specific targets in the system. And also this could be uh, as mentioned before, interesting insight that this could be a target of early stages of cancer, and is widely conserved among texts. So, well, in order to summarize the data we have been reviewing, as mentioned you before, there are two different well-known tumor processes in Tasmania devil, and these are this seems to different behaviors than the different behaviors that are too aggressive and are the main cause of the decline of this population. And for the purpose of identifying what is the differences between the devil facial tumor 1 and devil facial tumor 2, they develop a fast system, what is a reported system that let us to know what is the role of different proteins in the development of cancer and tumor processes. And through this technique, uh, they actually discovered that there are different processes of devil facial tumor 2 that is different for the type 1. And this is also regulated by the CD200 pathway. And this pathway is also well, well conserved among taxa. And also this could be a way to try to improve the overexpression of this pathway. Maybe could let us to know, let us to improve the, or how the boost the population of devil, Tasmania devil. And also this could be let us to understand how the different processes of cancer have evolved in different organisms, and taking advantage of this system that is also happen in wildlife. We know we can get the big picture of what is actually cancer processes and tumor process, and how is the behavior these processes in wildlife, and they and how they could let us to understand better the processes in human and in other species. So anyone have a question?
So thank you, Aranzazu, for an incredible paper presentation. So this, in fact, is really a very interesting paper because, as the authors mentioned in the introduction, like most of, and as you said also, that most of the immunological and molecular biology studies are focused mainly on these models animals that we all know, like mice, flies, human. But with this is actually with this strategy, what it allows to basically to target any type of species. So it's really a unique system, allowing all people that study really in the wild immunology or other different animals, allow the identification of any given protein, at least at the surface of the cell. I don't know, Patrick, if there's any way to adapt this to somehow detect intracellular proteins with this system, at least.
So at least for the one that we've used here, it's mostly been focusing on soluble and transmembrane proteins. But there is potential that you could use it for cytosolic proteins as well. It's just a matter of being able to make sure that you get clear signals from your fluorescent reporters and such, and being able to sort of isolate the receptor ligand interactions would probably be a little bit more difficult. But it has potential, and you could use other technologies that we've been developing alongside this, using nanobodies to probably help delineate those interactions too. Does that answer the question?
Yes.
Yeah.
Thank you. So I don't know if anyone in the audience, so in a couple of minutes, I think Dr. Andrew Fleiss will be joining to this session. So
I have a
Yeah, I have a comment or a question. Like, hi Patrick, I'm Emmanuel from Mexico, and I want to know if you proved the constructions of CDLA4 or CD200R with its transmembrane or segments, like studying this protein attached to the membrane and its interaction with its counterparts in our cells. Did you evaluate this, or only soluble proteins, fast proteins?
Mm-hmm. Yeah, cheers, Emmanuel. No, it's good. So typically the way that we set up the system was that either you would have one cell expressing a component, say the receptor, and then you would screen that against the soluble versions of the ligands that also have their own fluorescent proteins. So you'll have one stationary, so to speak, protein, and then you'll be able to screen the others against that. Makes it quite easy for flow cytometry to be able to just check that you've got both fluorescence signals in the same cell or not, essentially. So yeah, that was how most of the system was designed. There was also another version where we took the two different types of cells, because when you're doing flow cytometry, you can actually look at cells that have bunched together in your preparations, and you can analyze if it's because of those fluorescent signals or not. So we've kind of done both here. In terms of looking at between soluble protein and soluble protein, that's a little bit more difficult, and you need a bit more of an advanced setup. So this one we've kind of just designed to be able to go with the basics that most labs, well, I say most labs, but that that we would hope that most relatively decent labs would have in terms of like flow cytometer, cell culture, microbiology department, that sort of stuff. We've made it to be pretty much just a woe-to-go process, so to speak. Does that answer your question?
Yeah, that's right.
Excellent.
And also, I read the paper of bio protocol. When you describe the entire FAST protocol, and you say, well, your lab say that in that paper that yeah, you can also can construct fast proteins for transmembrane proteins, or for plasmatic membrane proteins. And I want to know if you of anyone in your lab worked with that type of proteins, like membrane attached proteins, and if do you evaluated possibly that this system is enabling the production of proteins that are indeed functioning like in the cell, that are attached up to membrane, and also can induce the interaction of two cells by this protein interaction.
Okay, if anyone had investigated cis protein interactions, so proteins on the same cell interacting?
No, in different cells.
Okay, on different cells. Well, yeah, I guess that carries on from the first point. Yeah, so essentially that was how our system was kind of designed to work. I think it's in figure, I'll pull up the paper now. In figure 4 of the paper, we sort of discuss how you can take the two different populations of cells and mix them together, and see if you get any crossover. So you can separate your cells that have just the one fluorescent color. So in this case, it's a 41BB with M cherry on it. And then you've also got all your other lines that have M citrine attached to their proteins. So with 41BB ligand or with CD80. And then you're just essentially mixing those cells together and seeing if those two populations of cells come together as a third population that has both of those fluorescences. And from that, you sort of, yeah, that's how you interpret the binding signal between the two, at least in this system.
In the co-cultured cells, you use this type of construct, membrane.
Yeah.
Okay.
Exactly. Though also on your other point, it was we also had with, say, like CTLA4, we were investigating that both in terms of its transmembrane ability, so having that expressed in the cell and just on the surface of the cell, but we were also investigating it with different tags. So for some versions, we made it without the transmembrane region, and so we could use it as a soluble version itself, and that's how we could sort of get the two sides of the receptor ligand interaction if we were only expressing like one cellular version and one soluble version, so to speak. Does that make sense?
Yeah, that makes sense for me. And I also have a naive question. There is only three type of cancer that are transmissible between in one species. I read that only in these the devils and also in dogs. These type of cancer that is known until the date uh, are transmissible only between from these species, but there is also a transmission of. Do you know if there is also transmission of this type of cancer between species, or are only found in uh, members for a specific species?
To my knowledge, it's only we only really know of transmission within species. Even then, the amount of transmissible cancers is only really limited to the ones in dogs, the ones in the two different variants that we have in the devil, and also there's a variant in soft shell clams, which is an interesting one as well. But yeah, in terms of interspecies, actually, no, I'll come back to that. The soft shell clam version is probably the only interspecies transmissible one that they've been able to find, and that's thought because of the way that these soft shell clams actually, reproduce and have sort of a spongy matrix that they're able to disperse their cancer a lot easier, and they have a lot stronger drive behind the cancer development. So yeah, that's the only one I know of that is interspecies. It's still only within the sort of other clam varieties that it spreads to, but that's the limit of my knowledge on that topic. The rest of them, as far as I know, are all just within the species.
Oh well, thanks for your answers, Patrick.
No problems.
So, Patrick, I have another question regarding Emmanuel's questions and the construct. So, I don't know. I'm thinking about because I'm thinking about some like classical microscope experiments. So, I don't know if this could also like if you could do Fred, I don't know, it's Fred experiments, which like if two proteins are close together, you will see a signal. I don't know if this could also be adapted to this model. It's Fred, I think.
I mean, there is potential. Though with FRET, you generally need to get the proteins. Like I think I can't remember the bounds of it. This is my neuroscience class is coming back to haunt me. I think it was within 3 nanometers they have to come in. So you either need to have a high enough concentration to attain that, or to have a very stable interaction that you know of. And that would have to be expressed probably within the same cell as well. So there is potential to do that, but you would have to be focusing on those interactions that are happening like intracellularly. It might take a little bit of modification of the system that we currently have, but I think there's potential there. Yeah, that's kind of how we made the system to be able to be like changed modular. So yeah, I think there's potential that we could investigate something like that. Did you have like an interaction in mind?
No, I'm just guessing. Like, I'm thinking of like all the possibilities you can do with this system regarding that. There's like really no much as I said before, like molecules or reactives to work with any type of animal. So I'm thinking that you could, with this particular system, you could actually expand with all the animal kingdom, if I could say that. But I really found interesting this work.
Well, yeah, I mean, it is built as a mammalian sort of based system. They're expressed in the Chinese hamster ovary cells to sort of get all the mammalian post-translational modifications on the proteins and to sort of model that. But there is potential as well to use it in other lines. I can't see why you wouldn't be able to modify it and tweak it just to suit your purposes.
Yes, thank you, Patrick. So think we will. So I think we will. I don't know if guys have more questions. I think we're about to expect to
Okay,
Andrew. Yes.
there is actually two questions of Alison Strangberg.
Oh yes, it's really a big question, so I'm gonna read it. Patrick, also if you could also check it on the chat. It says Alison Stanberg. She said, "I'm interested in what you believe that."
Eugenio, but
Yes?
we were waiting for Andrew.
Okay, sure. So I think Patrick is now. Are you still in Andrew's lab, or you moved to another lab?
Yeah, so it's actually been a while since I worked with Andy. I've been off in Europe doing my master's in infectious diseases, and only recently returned to Australia. But yeah, so it'd be good to see if I can work out some collaboration back with the lab before I head back to Europe.
Yeah, I was looking at. I mean, I noticed on your Twitter account. So are you still working with immunology, or do you just move to another area?
So I like to say that I'm sort of associated with it. I don't work explicitly in, say, transmissible cancer so much anymore, but I do work sort of in the field of stem cells in sort of the wider species models, and how we can manipulate adult stem cells to secrete antimicrobial peptides and other cool things, and how we can use that to our advantage, essentially, to fight diseases.
Wow, really? That's really interesting. So you now move to like the stem cell biology field, but like immunology, I think, or I believe that is somehow related with most of, like, if any, topics in biomedical science. So I think it's really good background to be an immunologist.
Oh yes, definitely.
Yes.
It is the field to be in right now.
Yeah, because yes, sure. In immunology, you can work with molecular biology, epigenetics, cell biology, differentiation. I think it's really a good model, and I think there's a lot of potential to expand it to any in other species. That I think it's really. I'm really happy to be an immunologist myself.
Exactly, the same here. I'll reach out to Andy and see if he's far off.
We're waiting for Andy.
So Patrick, you did your bachelor degree at the Andrews Lab.
Yes, so I've bounced around quite a bit actually. I started off in chemistry and neuroscience, and then I started volunteering with Andy's lab, and quickly discovered a love of immunology. And since then, I've just been bouncing around and changing my mind every 6 months. But I've always kept sort of close ties with the Devil Lab.
Yeah. So for sorry. Let me see. There's hold on. I think I don't know if I can.
I've just shot Andy an email. I think he had the wrong time for the questions.
Oh, let me. Thank you, Patrick. So So guys in the audience, we invite you to share this initiative with all of your friends and colleagues from class, and also to join us this Saturday to the Public Journal Club, the first Public Journal Club in developmental biology. It's going to be a really great opportunity to discuss Fernando Camargo's lab new paper. So I hope, and also guys, if you wanna, if you're interested in some topic, because now we're only having immunology and developmental biology, but if you guys are thinking of organizing like, I don't know, like any other topic, you can also reach to us so we could help you to organize. And we want this initiative to grow to other fields of biomedical science. So so we will wait just a couple of minutes to Andrew. We're really sorry for this. I think we have a problem with it at the time, but we hope we have Andrew.
Eugenio, we have a message in the chat from Jenny.
He says, Jenny, I have just started with the same lab as Andy. I think everyone's expecting the discussion at 9:03 a.m., but it's only 8:30 a.m. here. Oh my gosh. So I don't know if there was a, let me see if there was a problem with that. Oh my God. We scheduled like 7:30 a.m. East. I don't know what time is that, but supposedly was that day.
There are 15 hours of difference between Mexico and
So yeah, I think this was
Australia. So
yeah, because
yes,
yes, yes, I think because also Amanda and Andrew are like posting on Twitter. They're like retweeting our posts. So I think there was a confusion, I think. So I don't know. Yeah, okay, so so Patrick, do you like, or Jenny, do you know if there's gonna, if they could join, or we might, you know, like wait until 9:30 Australia? Or Patrick, what do you think?
Usually he's pretty good on answering his emails, so hopefully he'll get back to me in the next few minutes.
Okay.
But yeah, I wouldn't expect everyone to hang around till 9:30. But yeah, hopefully we'll get this sorted.
Eugenio?
Yes.
Yeah,
yes,
we have a new message.
Hey, Jenny, we have a team meeting in 9 a.m. 20 minutes, so we could tell them you're waiting. Oh yeah, I'm sorry. So we have this.
So if anyone have a problem to wait, maybe just we can wait until you join us. If anyone have to go in the audience.
So I hope we could all guys stay, because this time we'll have most of the lab, so it's really a good opportunity. The first time we have this in our public journal club, so I'm sorry for the inconvenience, but it's worth the time and effort. So, yes.
So,
but- I don't know.
okay. It seems that anyone have any problem to wait Andrew.
Yes.
So we just can wait for him and his group.
So Patrick, you are now in Europe or in
No, I'm back in Australia now.
Australia.
You know, with the state of the world at the moment, I figured it was the best place to return to.
Yes.
So yeah, I'm currently on lockdown.
So how's everything over there?
In Australia, it's all right. We're going through a bit of a second wave at the moment in Victoria and New South Wales. But most of the country's been able to lock down pretty well, and we've got it under control for the time being. But yeah, hopefully I'll be able to return to Europe sometime in the future.
But you managed to finish your master's, or you are still like online?
My thesis is due in a week.
Oh my god!
So my project was mostly lab based, and unfortunately that had to be entirely switched over when they closed the labs. So now it's all just a literature review.
Okay. So I think. Oh, wait a second.
Where are you now?
Yes, I think we have Andy now. Yes. So hi, I think Andy is really now. There you go, Andy. Hi, Andy. We're really sorry for this time inconvenience. I think we have a problem with that, but thank you for joining to this Public Journal Club sessions. We're really excited to ask you a couple of questions of this great work. And once again, thank you for being here, and really sorry for the inconvenience of the time.
Yeah, I apologize for being late. Sorry.
Don't worry. So, so just give me a second. I will be introducing Andrew. So Andrew, I mean, I just got your resume, and I think it's really impressive, all the things you have done. So you started as a computer science, right? So Andrew Fleiss studied computer science in Minnesota State University, and then later he moved to Johns Hopkins University, and then finally to Michigan State University. Well, he did a dual PhD in zoology and ecology, evolutionary biology, and behavior. Then he moved to, I think, to several postdocs until he finishing in the University of Tasmania, and he has almost 29 peer-reviewed publications and several fellowships and grants received through his career. So it's really an honor that Andrew being here in the Public Journal Club. So if anyone in the audience wants to ask any comment to Andrew related to his paper.
I'll keep an eye on the chat too if anyone wants to type them in, but otherwise happy to have people blurt out their questions.
Okay, maybe you want to ask the questions on the chat that
May I ask?
Yeah.
Thank you very much. I would like to thank you for Public Journal Club for inviting us. And also I want to make some questions for the principal authors. I think that questions that would be interesting for me. I have to accept I was not really familiar with transmissible cancers. So right now I would like to ask, well, how could two transmissible cancer emerge in the same species? And the second question would be, how will the two DFT variants evolve? If you want to contribute with an answer, I will be delightful.
Yep. So the first transmissible cancer was found in Northeast Tasmania, and it was actually a photographer that was just out in a wildlife park and took a picture and saw a devil with a strange tumor on their face and didn't think much of it. And then over the next 5-10 years, they started seeing more of these strange disfiguring facial tumors. And then around 2003, the first devil was trapped with this tumor. And they looked at the cytogenetics of the tumor, and there had been major chromosomal rearrangement. So devils have 7 pairs of chromosomes, and some of them have clearly been pieced together in different places, like parts of 3 were on number 1. So that suggested something strange was going on. And then they would see the same cytogenetic rearrangement in every cancer. And when they started sequencing the tumors, the transmissible cancers didn't match the hosts they were on. So lots of differences. There'd be somatic mutations that didn't match between the tumor and the host. MHC type between tumor and host wouldn't match. So there was a 2006 paper in Nature suggesting that this was actually an allograft. So the tumors are transmitted between devils by biting. So that tumor, first found in 1996, has now spread across almost the whole state of Tasmania. So I don't know where you're all from. Tasmania is probably about the size of Switzerland or Iowa, for those of you North American and European people. So it's spread across most of the state, and it's killed thousands of devils. The population was estimated to be over 100,000 about 20 years ago, and now it's probably 20 or 30,000. And that reduction is primarily due to this first transmissible cancer. In 2014, Ruth Pye, a veterinarian from our group, was called out on a welfare call. They saw a devil with a big facial tumor, so she went out to euthanize it and took samples. And then when they did, they found that it didn't have the diagnostic marker we used for this first transmissible cancer. So the first transmissible cancer arose from a Schwann cell. So it very highly expresses periaxin, which is part of the myelin sheath that surrounds peripheral nerves. And this transmissible cancer that Ruth found didn't have this. So they started looking at the cytogenetic patterns and single nucleotide polymorphisms, and it turned out that this was a different type of transmissible cancer. So this is really bizarre. The only other known transmissible cancer in mammals is in dogs, and that's a sexually transmitted cancer. It's been going for about 8,000 years, and that one's really strange. It's dogs have 78 chromosomes, and that tumor is usually found with somewhere between 56 and 64 chromosomes. So it's just been shedding chromosomes over all this time. So the fact that devils have 2 of them is pretty worrying. It means they may be prone to these types of cancers. Both of these came from Schwann cell precursors, so there might be something to do with their Schwann cells. And then the big question for our immunology team is, these tumors are genetically mismatched between the hosts. They have differences at MHC, they have different somatic variants. So it should be rejected by the immune system, but it's not. So that's one of the reasons we're focusing on the checkpoints as we did in this paper. And any of you that study comparative immunology know there's usually not a lot of reagents for comparative immunology of oddball species like this. So we decided to make our own reagents, and it took about 5 years to get this system fully up and tested, but we're pretty happy with it, and we can do a lot with the system now.
Thank you very much, and congratulations about your paper. It's really, really interesting.
Thank you. Looks like there's a question in the chat. I'll take that, if that's okay with the people running. Okay. The question is, I know this is not a very related to the paper, but can we know the story behind the Twitter profile with the leopard? For my PhD research, I spent about a year living in Kenya. I lived in a tent in the Masai Mara on the border of Tanzania and Kenya, which is absolutely fabulous place to visit or do research if you ever get the chance. But my daily life there involved waking up about an hour before the sun came up and going out with radio tracking equipment and finding spotted hyenas. We had recorded all their behavioral interactions in the morning and in the night. And these are big groups of hyenas, about 100 in the clan. And we'd have to sometimes identify 10 or 15 at a time. And during I was studying them because hyenas get rabies, canine distemper, anthrax, and they don't die from it. So I was interested to know what their immune system is doing differently than other species. Short answer there is I did not answer that question, but we did some good basic research on the spotted hyena immune system. The picture in the profile with it's actually a cheetah in the background, is we were out looking for hyenas. excuse me, and 3 male cheetahs decided to climb up on our research truck and use it as basically a rock to scan for prey. So we had about 45 minutes where there was 3 cheetahs sitting on our truck. We eventually had to chase them off because they tried to chew down the antenna that we use for tracking hyenas. So it was a pretty good day in research. I haven't had one like that in the lab yet.
So I think we have a question. It's really a big question from Alison Sandberg. So it said, I'm interested in what you believe that a million years ago of conversion implies for transmissible cancer over story. Natively, I would think this implies that they are rare, since if they were common, you will expect that to drive an arms race of CD200 evolution. But the existence of 2 separate strains rising in 30 years in Tasmanian devils could imply they are relatively common. So I don't know if you can read this, ask these questions, but if not, I will copy again to the chat. So put
Yeah,
it
if you
here.
could copy it to the chat,
Yes.
that would be useful.
Yeah,
I
because
think
it's really
I got,
a big, it's a really big
yeah.
question there.
Yeah, you believe 160 applies for trans. Yeah, so I'll step back and talk about the transmissible cancer issue again first, and then get into more of the checkpoints and the CD200. So with the transmissible cancers, I said there's the 1 in dogs and 2 in devils. There's been 5 discovered in marine bivalves now, so clams and mussels. And some of those transmissible cancers actually cross species. Obviously, clams don't have the type of adaptive immunity and MHC recognition that vertebrates have, but they do have an immune system. So crossing species is a major thing there. Given that all of these 8 transmissible cancers have been discovered in about the last 15 years, I think there are probably more of them out there. But the thing that works for these is the transmission mechanism is there. So with the marine bivalves in the ocean, the leukemias, the clam leukemias, basically just get out into the water, and these bivalves are filter feeders. So they filter in the transmissible or the leukemia cells, and then they get infected and get cancer that way. For devils, it's through biting. So devils are always fighting and biting each other. So there's a transmission mechanism there to keep it going. If I had a transmissible cancer on my face, it wouldn't go anywhere because I don't think anybody's going to come along and bite me on the face, maybe my 2-year-old or 5-year-old, but the transmission mechanism's not there. The dogs are an interesting one because it's sexually transmitted, and certainly sexual transmission is an option, or is a good transmission pathway for a lot of diseases. The trick there is that if it originates somewhere else in the body, it has to be able to migrate to the genitals initially to start that transmission process and then be able to keep going that way. So those are some of the reasons why we think there's not more discovered and more known. I'll also say that for human medicine, there are cases of essentially transplanted tumors. So one of the most interesting cases is that there was a man who died of ovarian cancer. So he got a transplant from a woman that had died. I don't know if it was a kidney or a liver or something, but there was metastatic ovarian cancer in that tissue that he received, we'll just say kidney. And then when that got into him, it metastasized throughout his body, and he had ovarian cancer. And there's other instances where 4 different people got a metastatic cancer from transplants that all came from the same person. So it can happen, but the transmission, you have to think of it as more of an infectious disease in some cases. As for the CD200, that's an interesting question too, because it's a lot of these immune checkpoints, they have multiple receptors. So I think the one that a lot of you are probably familiar with are PD-1 and its receptors PDL1 and PDL2. Those are the ones really making big waves in the clinic, and PD-1 antibody is predicted to be the top selling drug in the world by 2023. And that has 2 different receptors, PDL1 and PDL2. CTLA4 and CD28 both bind CD80 and CD86. So we think the way that, well, this is not my hypothesis, there's been some very good papers on it. I think Barclay, B-A-R-C-L-A-Y, is one of the authors on some of these papers, but they suggest that these stimulatory or inhibitory pathways come about usually from a 1-to-1 receptor. So say CD28 initially was just binding to CD80, and at some point early in vertebrate evolution, CD80 split into 2 different receptors, so CD80 and CD86. And in that case, it's not obvious that one's inhibitory and one's stimulatory. But for some of the other receptors, there can be one that gives a positive signal and one that gives a negative signal. And the hypothesis is that it's basically the immune system evolving to try to mitigate risk of a pathogen hijacking a certain pathway. Viruses, I think there's a herpes virus that uses CD200 or CD200R to actually enter cells. There's another receptor called herpes virus entry mediator, HVEM, which binds to BTLA4 and I think CD160 is another name for it. But there's a lot of pathogens evolved to use these checkpoint receptors. So a lot of them have actually evolved to have an inhibitory and a stimulatory paired receptor. And so I think that's kind of what this question gets at, is that if a certain immune checkpoint such as CD200 is exploited by a pathogen, then we might expect that there's actually some evolution on that receptor, either for different binding or perhaps duplication of that receptor. So sorry, that was a bit of a wordy and rambling response. If you want more detail on a specific part, just let me know.
I think it was perfect. So I did have a question related to if there's any. I mean, I don't know much about this topic, but if there's like any correlation. I mean, at least in humans, but that you can also bring back to your study. Like, there's any correlation between checkpoint inhibitors or like mutations or something and MHC molecules related to the development of cancer. So because I'm also thinking there's only like MHC molecules mutation that would lead somehow to development of cancer, but I don't know if there's any study related to the checkpoint in like I don't know, like polymorphisms or something to that. And my second question is like more related to the animal model. So I don't know if you could, like, if there's a, first of all, I don't know if there's a cell line or that you could use for inducing a tumor model in anatomic mice using Tasmanian devil transmissible cancer. So you could study like like in the development of the tumor in the mice with the
Yeah, so I'll do the first question first. So I think MHC is highly polymorphic, and that's one of the ways that you distinguish foreign tissues from coming into the host. So it's an interesting question, and something we're actually looking at in human checkpoints, and not necessarily polymorphisms, although there are single nucleotide variants that play a big role. So for CTLA4, that's a powerful immune checkpoint protein that circulates from the cytoplasm to the surface constantly. And there's an associated gene, cytoplasmic protein, that regulates that transport to the surface and back to the cytoplasm. I think it's called LRBA. And a mutation in that disrupts the function of that causes a completely different CTLA4 function. So that's a case where polymorphism is clear. And I think one of the benefits of looking at comparative immunology in devils or dogs or whatever species is you can look at some of these polymorphisms and see how the function actually does change. So in the rewilding immunology paper that we just had published in Science, I cited a paper from pigs from around 2000 that pigs are one of only 3 mammal species out of about 140 that have CTLA4 sequence so far that have a different receptor binding domain. So they have an leucine substitution for methionine. So that's a pretty interesting change. And for me, it raises a question as if pigs are good models for xenotransplants, because they are actually one of the most common large animal xenotransplant models. What we've been doing in our lab with the human checkpoints is looking at, excuse me, basically alternative splicing of these. So you can have the same genes, but sometimes an exon gets skipped or an exon gets picked up in a different region, and you have a different form of that. So we've went through about 40 different human checkpoint isoforms and looked at the binding to receptors, and hopefully we'll have that submitted pretty soon, hopefully up on bioRxiv within a month or two. But it's definitely an interesting thing to look at, and looking at other species provides another way to dig into that question. As far as the transmissible cancer, basically xenograft model, so that has been done. So we have dozens of these transmissible cancer strains that grow pretty well in the lab most of the time, and our group has transplanted them into mice before, and we can get them to grow in nude mice is usually what we use, and they grow pretty well. And then we can test treatments there in a more in vivo model. There's a group in Austria led by Andreas Bergthaler, and they've used these mouse models to test different tyrosine kinase inhibitors. And some of the RTK inhibitors have done pretty well at arresting tumor cell growth and enhancing MHC upregulation, because the transmissible cancers, at least the first one, generally doesn't express MHC, but some of these tyrosine kinase inhibitors can help upregulate MHC or potentiate MHC upregulation in combination with interferon gamma.
Okay.
Um,
Thank you, Andy.
hey, hi, Andy.
Hello.
Aranza. And here, Aranza. And just, I was wondering, well, there is a well-accepted thought that the fixation of MHC alleles is a way to avoid the population decline, not only in Tasmanian devils, in other species also. And I was wondering how MHC upregulation by CD200 or CD200 R1 pathway could solve the decline of this population, because I think the overexpression of these MHC alleles could also drive the decline of these organisms by the autoimmunity, isn't it?
Yeah, so I think the way that I would envision the CD200 being a target for helping keep the devil population stable is that if and this is based on some trials that have been done in dogs, and there's a phase 1 human clinical trial coming on for CD200, is that if you can block any of these inhibitory pathways that seem to be helping the immune evasion of the cancer. So if we had a CD200 monoclonal antibody or peptide inhibitor, this might allow cells such as NK cells to start recognizing the tumor, because the tumor cells should be expressing MHC, and if not, then the missing self hypothesis suggests that NK cells should start attacking those. And NK cells can be a pretty good source of interferon gamma, so that could lead to upregulation of MHC on the tumor cells. So with the CD200, I mean, that would be a more immunotherapy pathway, and that has limited value as far as conserving the whole population. So we could trap devils and bring them into captivity and do immunotherapy, and that is one of the things we're hoping to do. But we can't trap every devil across the whole state of Tasmania. So our primary immunology-based conservation priority is to develop an oral bait vaccine. So we actually use a viral vector, and we're focusing on adenovirus that could encode some devil facial tumor antigens, and then also some potential immunomodulators, so interferon gamma or a CD200 nanobody, so that this vaccine would then be packaged into a bait. So something the devils want to eat, they bite into it, the viral vector spills into their mouth, and they get immunized that way. And that probably sounds like a pretty crazy idea. We have published this proposal in Expert Review of Vaccines, and this approach has been used for vaccinating wildlife against rabies for more than 50 years. So it's eliminated rabies from a lot of Europe. It's controlled. It's been used on 4 different continents. And in the US, one of our collaborators is distributing 10 million to 20 million oral rabies vaccine baits across the eastern seaboard of the US and in Texas and other parts of the Southwest. So that's how we're envisioning doing this. And basically, the more we understand about the checkpoints and cytokines, the better we could make this vaccine, and the more precisely we could target the immune response towards the tumor cells. Did that get at the question, or was that answering something else?
Yeah. Oh, thank you.
Yeah. I see a question here. I'll jump to that if that's okay. It looks like it's from Diego Perez Vasquez. I was curious to know if this method can be used for other techniques besides flow cytometry, such as fluorescent microscopy. I'll say definitely yes on that. And our method is, it's not completely new. So people have been fusing proteins to fluorescent proteins for a long time. And a lot of our other studies, we're actually using surface proteins that have the fluorescent protein in the cytoplasm, and we're using this to track cells or proteins being transferred between cells, so through trogocytosis or transendocytosis. We have a paper on bioRxiv right now that looks at protein transfer of CD80 and 86 by CTLA4 and CD28. And because we're usually, our primary method is flow cytometry, so we've focused on that, but we've worked with some neuroscientists and other people that are much better in fluorescence microscopy, and you can see these proteins on the surface if they're fused to a surface protein. Once they get secreted out of the cell, they're harder to see, but if you do a long enough co-culture experiment or staining experiment, you can actually label another cell and you can see them. I'll also say that most of these soluble checkpoint fast proteins that we make are generally monomers. So because it's a receptor ligand interaction and it's a monomer, the affinity is generally pretty low. So we've used the FC tag to form a dimer, and that gives a bit better avidity. But one of the other things we did in the paper was fuse the fluorescent protein to a nanobody, and this works really well. So you could actually do a co-culture with the nanobody, and because those have much higher affinity for their targets, you can see them much better in fluorescent microscopy that way. Just one more question here. Did I work in mice in UCL? And I'm very interested in this paper, in particular, how much we are missing by focusing only on mice. What would be your suggestion to promote a more broad research on immunological and molecular mechanisms going further than lab-based models? So what we've kind of proposed in this other paper that just came out in Science is that we don't want to say anybody doing mice research is wrong. There's been so many great discoveries come out of mouse research, but what we're saying is that there's great opportunity to start studying other species. And I think the simplest way forward with this is to just start studying mice that are taken out of the lab into a more natural environment, or studying wild mice. And there's been some really high profile papers on this in recent years, with one of the first ones being in Nature by David Mazepus group at the University of Minnesota. So they just took your regular lab mouse, and then they went down the street into a pet shop and bought some mice at the pet shop, and they housed them together. And the immune system of the lab mice just changed immediately just from this interaction with the microorganisms that inhabit the pet shop mice. So that's a really simple way to do it. Another way that's been quite productive already is to just take the lab mice and house them outdoors. So Andrea Graham at Princeton is leading some of this work, and there's some accepted differences that some types of mice can get more parasites, or a lot of people know the Th1, Th2 paradigm that was first discovered in BALB/c mice. But what she's finding is that when you take these strains of mice and put them outside, in the dirt, in the grass, where there's potentially parasites, these differences between B6 and BALB/c and CBA3, they start looking less like differences because they have a normal microbiota. So that's the simplest way to do it. And the other thing I'd advocate for is when you have a specific disease, particularly one that's relevant to domestic animal production, cows, pigs, chickens, or dogs, or to Tasmanian devils with a specific disease, is you take these very interesting diseases or economically relevant diseases and try to do a real in-depth immunology study. Approach it as a mouse study, which means you need to develop all the reagents first. But if we can start doing some really in-depth studies in these other species, I think it will catch on more, and we'll start finding some really interesting things. And one of the examples we pointed to in the other paper is that nanobodies, or single domain antibodies, were discovered from studying antibodies in camels. It was that simple. They looked at the antibodies, they looked strange, they followed up on it, and now nanobodies are one of the most effective tools being used in studying the COVID and the SARS-CoV-2. So you don't know what's going to come out of these comparative immunology studies, but it can be some really exciting things.
Hey, hi Andrew. I have a questions about your about the fast method. Well, I want to know if cell transfected with these fast constructions are stable, or permanently expressing the fast constructions? Or do you know? Do you prove it in the cultures, in the transfected cultures, if the cells are permanently expressing these proteins?
Yeah, so good question. So the system we're using is the Sleeping Beauty transposon system. And these were believed to be pretty stable because it's basically like a jumping gene or a retrovirus where it has inverted terminal repeats on each end of the cassette you're trying to insert. And then when the transposase is there for the Sleeping Beauty, it just goes and cuts open the DNA. And if I remember correctly, it's between an A and a T, but it could be between two T dinucleotides. But anyway, it gets inserted into the genome, and the vectors that we've made. So it has the insertion cassette, and it also has the transposase in the same vector. And the transposase is driven by a CMV promoter. So we think it's making a lot of the transposase. So there's probably multiple copies getting inserted into the genome. So it should be stable. We have another paper that hopefully will be submitted pretty soon from Chrissy Ong in our group. And she's looked at the stability of the transfection over about 3 months. So we select the cells with hygromycin initially to try to make sure that all of the cells are expressing the transgene. But then after a month of drug selection, she took the drug off for 3 months and the expression hardly went down at all. So it looks like it is a very stable transfection. And yeah, so I think it's a pretty stable transfection and it is integrated in the genome. And we've done RNA-seq analysis of these cell lines too, of the cargo that's inserted, and that's driven off a human EF1 alpha promoter, and it's the highest expressed genes in the transcriptome using RNA-seq. So we think it is stable, and we think it's driving really high levels of the protein expression. Oh, you're muted, Emmanuel. So I can't hear you, Emmanuel. I think you're muted.
Emmanuel, you're muted. Over.
Okay, sorry. Thank you for your answer. I want to know also if do you prove to transfect freshly isolated cells, and there is possible to, or do you have protocols to isolate immune system cells from the devils, and then transfect and then reinsert the cells in the devil, so to track the immune interaction of maybe these immune checkpoints in the
so that it's a really interesting question. We haven't done that yet. We've tried we've used this system on other cell lines like Jurkat cells from human, that's a human lymphoma, a T-cell cancer basically, and it didn't work great on those. It worked pretty well on K562 cells, which are a human erythroma cell line. We're pretty confident it will work on peripheral blood mononuclear cells isolated from devils or humans or whatever, because the Sleeping Beauty system is being used to make CAR T cells for humans because it's a very stable transfection. So it is being used that way. I think all of our transfections are done here just using polyethylene amine because it's the cheapest way to do it. But we're looking into electroporation now, which you get a lot higher transfection efficiency. And I've seen some recent talks about using nanowires and other nanotechnology to increase the transfection efficiency. So there's the possibility of that. I don't think we'll get to the point where we're reinfusing GMO cells back into devils. But for the xenograft mouse models mentioned before, it certainly would be useful to knock in or knock out some of these checkpoint molecules and then see what happens in the mice.
Okay, and last, instead of inserting fluorescent proteins at the end of the construction, like EFP and or not like M cherry, M orange, and all that, have you proved that to put another type of tags like I don't know, like maybe luciferase to make breath analysis, or create a system for two-component or for hybrid interactions between proteins to
Yeah,
measure
that's
interactions?
that's definitely something I'm interested in. We have some of our vectors that do express luciferase on this. It's not been fused to the tail, to the C term or N term of a protein yet, but we do use the system for expressing luciferase, and it works pretty well. As far as looking at protein interactions, there's been some really interesting papers on that recently, and ways of assessing that. There was one in Nature about a year and a half ago, and they called it the lipstick method, and they saw the proteins kissing across cells, and they would tag the cells that way. So that's a similar approach. I think what you're getting at here is like a split reporter. So like half the GFP fused to one protein and half fused to another protein, and then, or luciferase split. So you only see the bioluminescence or the fluorescence when the two proteins are in close contact. I am designing some vectors for that now, but I have enough other stuff on my plate and unfinished papers that I probably won't get to that soon. But I think that is definitely possible. And actually, one of the first study that turned me on to using this method is a paper, I think 2010, by Haas, Haas. And what that paper did is they fused M-citrine to recombinant antibodies, and they would put half of the M-citrine on different heavy chains. So they could have a bispecific antibody that bound two different targets, and half the M-citrine would be on one heavy chain and half would be on the other heavy chain. And then when the two heavy chains were joined through disulfide bonding, then the M-citrine lit up, and you know that you had your proper bispecific antibody. So it's a really interesting approach, and that's why I used M-citrine as the test case for most of my studies, is that it's a monomer, and I knew it would work in that system, whereas GFP usually forms a dimer or aggregate. So that's why I stayed away from GFP. But there are new versions of GFP now, the superfolder, and there's a monomeric ultra stable GFP that we'll be incorporating into some of these new vectors.
Okay, well, thank you for your answers, Andy.
Andy, I have two more questions regarding to the CD80 and CD86 expression, and I was wondering why the CD8 and CD86 expression is only upregulated in the wild devil facial tumor 1 and tumor 2, but only in the wild type version of these tumors, but not in the other tumors.
Is this the same question that Roman sent in, or is this a different one? Sorry, I was reading Roman's question and listening to you at the same time, and now I realize they might be different questions.
No, I mean, okay, I will share this my screen.
Oh, okay. So figure 5. Are you talking about figure 5A in the paper, Aaron Zazu?
Yeah.
Okay, let me just scroll to that figure to make sure I'm looking at the right one. Okay, so 5A.
Yeah, 5A.
Okay, just bringing it up to make sure my memories. So the question is, CD80 and 86 only upregulated in wild type DFT1 and DFT2, but not in the other tumors. I'm not sure. I don't have a great answer for that. The bits of staining we've done, it doesn't look like there's a lot of either of those on the protein by default, but there's some pretty interesting papers coming out recently that about the interactions with CD80 and PDL1 and PD1. So CD80, at least in humans, and I think in mice also, will bind to PDL1 in cysts. So when they're both in the same cell, CD80 and PDL1 actually bind. And what's really interesting about this is that CD80 binding to PDL1 blocks PDL1 from binding to PD1 in trans, so on a different cell. So a tumor, basically a tumor that expresses CD80 and PDL1, the PDL1 won't bind to PD1. So that's quickly getting pretty complicated. But the CD80, even when it's bound to PDL1, can still bind to CD28 and give a co-stimulatory signal. So this hasn't been looked at that much in tumors. There was a paper about a year ago, I think in, I'm gonna get the paper wrong, so I won't say it. but suggesting that some tumors do use CD80 as a potential immune evasion mechanism. But I think the depth of knowledge in that area is really shallow right now, and I don't have a good answer for that yet. So it's a good question, but I don't have a good answer for that. I guess just that it's complicated.
I don't know, can I ask you one more question, Andy? I was thinking about the data of circulating tumor cells, and the fact that the Tasmanian devil is not that polymorphic, so that's why it's able to transmit. So, is there any report of transmissible cancer between mother and like the pup?
Yeah, really good question. So that's one of the big worries as far as conservation goes, because if to date we haven't seen evidence of transmission of the tumors from mother to the joeys, and it's a very close, intimate contact for a long time. So these devil joeys are born very precociously, and they live in the mother's pouch for quite a while, but they'll come out and they'll certainly have social interactions of licking on the face, but there hasn't been evidence of it being transmitted from mother to the offspring. There was one case in captivity where a mother developed the tumor. She was brought in and they thought she was tumor free, and eventually a tumor showed up. And then like 14 months later, I think one of the joeys showed up with the cancer, but they're not sure if that's because they had interacted with other wild devils through a fence and picked it up that way or not. But if the tumor does start doing vertical transmission, that creates a much bigger problem, because this population has been supported in the wild basically because all the adult devils die. So for most of the last 20 years, every devil over 3 or 4 years old was, well, they didn't get to 3 or 4 years old because they died because of the tumor, but there was always enough new offspring coming in to maintain a low level of the population. But if it started doing vertical transmission from mother to offspring, then that might create a problem for maintaining the population. And given that mothers and offspring would have shared MHC type, that might actually make that easier to happen. But so far, it doesn't look like that happens, and we don't know why. Maybe some differences in the developmental processes of the immune system in devils.
Okay, thank you. I don't know if you are like okay with the time, so if we could go with more questions or.
Sure. Yeah.
Sure.
This is fun.
Okay. So, okay. So I think we have some in the chat. I just want to like one more question. Do one more question regarding like it was very interesting. The first question that I think someone in the audience did, that like I was thinking that how feasible is this to happen again. So imagine that we could, like, the scientific community could eradicate the tumor devils, not like the tumor devil. Yeah, this transmissible tumor in the 1 or 2. So you have now a population with no tumor at all. How feasible, or like the probability is that gonna happen again in this type of population? So I'm thinking about that like people that do conservation, they have to look more now into this immunological data for developing or strategies for these a particular species.
Yeah. So the question of trying to eradicate these, I'll say my view here, and this is certainly not shared by all people that do this type of research in devils or other species, is that we're trying to develop this oral bait vaccine with the goal of probably trying to contain the second type of transmissible tumor. It's found currently only in southern Tasmania. So there's still potential to get that one before it spreads across the whole state. So we'd have to move pretty quick in our vaccine efforts. So my goal is to try to eradicate that. And if we have a bait that we can spread across the landscape pretty widely and vaccinate that way, and it is protective, then I would say there is the potential for eradicating this tumor. Doing it across the whole state, that would be a pretty monumental accomplishment, and it would require a lot of funds. If, back of the envelope calculations, if it was $1 per bait vaccine and you had to cover the whole state, it would probably cost $5 or $10 million. So the question comes up, is it worth trying to do that? And then I would say yes, because there's a lot of funds and going into trying to save the devils and help save the devils right now. And the longer these tumors are around, I think the more it's going to decrease the general genetic diversity and long-term ability of these devils to persist long-term in the environment. So I think it is worth doing, and our group is pretty motivated, and we have a really good team that has the skills necessary to do a lot of this stuff. So we're vigorously pursuing this, but it's with the idea that the system could also be used for other diseases and other conservation purposes. So we have a grant pending right now to develop the bait vaccine for devils, but also to test a bait vaccine for rabies. So we currently don't know if rabies infects Australian marsupials, and if rabies came into the wild dog population in Australia, we haven't tested a bait vaccine here. So we're working on that in parallel. But a similar approach is being tried for vaccinating prairie dogs in the USA to protect them against plague. And protecting prairie dogs is one thing, but they're the main prey species for black-footed ferrets, which are an endangered species over there. So it's an interesting concept that you're using an oral bait vaccine to vaccinate a prey animal so that the predator can kill and eat that animal that you've saved. And the ferrets can also get the plague, so it could protect both of them that way. A similar approach is being tried for white nose syndrome in bats. So I think a lot of you are probably familiar with bat populations in the States have been crashing due to white nose syndrome, and in other places. It's a fungus that gets on the nose of bats, and then when they hibernate in the winter, the fungus just kind of keeps spreading, usually starts on their nose and then spreads over other part of their bodies in the winter, and it decreases their ability to thermoregulate, and then they end up dying from that. So one of the approaches being used there is basically fumigating a cave with a vaccine, probably a viral vector that stimulates an immune response, and then the bats would be able to fight off this fungus before it takes them out in the winter. And there's a couple other, this approach is being used for bovine tuberculosis in badgers in Europe. So the culling of badgers or the control of bovine TB in the UK is a huge political hot button issue. And it seems that an oral bait vaccine to protect against this bovine tuberculosis is reasonably effective. So the similar approach is being applied to a lot of things, and I'll use, so I think it's a useful approach for conservation. So in Australia, there's been 100 species gone extinct since Europeans arrived in Australia. And a lot of those, some of those were associated with disease, and wildlife disease is becoming a bigger problem. And then the final point is that emerging infectious diseases, the coronavirus being the obvious example, these are coming out of wildlife. So if we could find potentially sources of pathogens that emerge from animals and go into humans, and if we had a wildlife vaccine that we could distribute widely, it might be a way of reducing the risk of emerging infectious diseases in humans. So I think there's a lot of good reasons to pursue this, some of them immediately practical and some of them more academic.
Thank you very much, Andy. So I think we almost run out of time. So there are really more questions in this topic, but I think people in the chat could also, I don't know if you're okay with that, send you an email or a tweet. It will be okay. Just before we leave, we want to hear from you a take-home message, not only related to your work to this paper, but related to immunology, basic research in wild immunology. What is the take-home message you want to all of the audience to hear from you?
I'll give a bit longer answer, and then I'll hopefully be able to distill it down to one sentence. So I started my research career in a very cutting-edge human immunology lab, and I learned a lot of the techniques, T-cell, using MHC tetramers for looking at individual T cells and making monoclonal antibodies. Then for my PhD, I went and studied spotted hyenas, and that was the far end of the spectrum. So instead of having every possible tool you could want for immunology, I had 0 tools. So that was pretty frustrating. So when I came to the devils, I decided I would try to make our own research tools. And it's a big startup cost now, or it's a big startup cost to do that for each species. But I think we have a very good group in place now with our team here studying the devils. We have veterinarians in the team, Ruth Pye, who I see is signed on, and we have Amanda Patchett, who's doing some fantastic bioinformatics work. And this Wild and Comparative Immunology Consortium that we've started has people, very good structural biologists doing protein crystallography, people studying fairy wrens up in the Kimberley of Australia. So we have a really diverse group of people. So my take-home message here is that if you have an immunology-related research question and you have a problem, write to us and we'll try to come up with a solution together, because the tools are in place, the technology is advancing, so we can probably get it done.
Thank you very much, Andy. So just before we leave, can we all guys turn on your camera so we can take a picture of the meeting? Just hold a second. So we have Carly. So Manuel, I think, or Aranzazu, if you could help me with the picture. I cannot take the picture right now, but so I don't think Carlos is there. Diego, okay. So okay, so just tell me, Aranzazu or Emmanuel, if you took the picture.
Okay. 1, 2,
yeah.
and 3.
1, 2, 3.
Okay.
Okay, so thank you guys very much for joining to this session, and congratulations, Andy, and all your team for all the work you are doing for conservation and all these tools you have developed. Thank you very much for joining to this session of Public Journal Club.
All right. Thank you for the interest in our research and chatting with us. Bye.
Yeah. Thank you. Bye.
Thank you, Andy.
Thank you. Bye.
Thank you.
Thank you, Patrick.